Good morning, students. My name is Sandeep Saini, and we are going to start the lecture of crop physiology having a course code of BSAG 204. Lecture name is seed structure. Okay, so in this lecture we have important topic is seed structure, their biological and morphological changes. So what is seed? The most important and basic term we have studied from our earlier classes at what is seed. A seed is a basic part of any plant. We can say a seed is a basic part of any plant. We can say a seed is a premature embryo. The ovule after fertilization develop into seeds. A seed is made up of a seed coat and an embryo. The embryo is made up of a radicle, an embryonal axis, and one or two cotyledons. A seed is found inside a fruit which converts into a new plant when we plant it. Hence, the seed is the most important part. We can see the diagram of a seed having a endosperm, epicotyle, hypocotyle, radicle, cotyledon and seed coat. On our right side, we have corn seed which is of monocot seed. On the left side, we have bean seed which is of dicot seed. Okay, now next is types of seed. A seed is primarily of two types. The two types are, first is monocotyledon seed, next is dicotyledonous seed. Structure of monocotyledon seed. A monocotyledon seed, as the name suggests, has only one cotyledon. There is only one outer layering of the seed coat. A seed has the following parts. The first is seed coat. In the seed of cereals such as maize, the seed coat is membranous and generally fused with the fruit wall called hull. Next is endosperm. Endosperm is a bulky and stores food. Generally, monocotyledon seeds are endospermic, but some are in orchids and non-endospermic. And next is alluronic layer. The outer covering of endosperm separates the embryo by a proteinous layer called alluronic layer. Next is embryo. The embryo is small and situated in a groove at one end of the endosperm. Next is scutellum. This is one large and shield-shaped cotyledon. Next is embryonal axis. Plumule and radicle are the two ends, which is called embryonal axis. So we can see here endosperm, coleoptile, plumule, radicle, endosperm, alluron layer and embryo. Next is the structure of dicotyledon seed. Unlike the monocotyledon seed, a dicotyledon seed, as the name suggests, has two cotyledons. It has the following parts. First is seed coat. This is also the outermost covering of the seed. Next is helium. Helium is a scar on the seed coat through which the developing seed was attached to the fruit. You can see the helium is this blue colored. Next is micropyle. Micropyle, it is a small pore present above the helium. Next is embryo. It consists of an embryonal axis and two cotyledon. These are often fleshy and full reserve food material. Radicle and plumule, they are present at the end of the embryonal axis. Next is endosperm. In some seeds such as castor, the endosperm formed as a result of double fertilization. It is a food storing tissue. Next are the changes during seed germination. The first one is physiological changes. Physiological changes, the first is water uptake. Seed germination starts with the imbibition of water by seed coat. Due to imbibition of water, the seed coat becomes more permeable to oxygen and water and less resistant to outward growth of embryo. After imbibition, the inner content of the seed increase in volume, thereby exerting pressure. The plumule and radicle emerge thereafter. वाटर अपटेक के टाइम सीड जर्मिनेशन में जब सीड जर्मिनेशन स्टार्ट होती है तो वो ड्राई सीड है इम्बिबिशन करता है वाटर की. ड्यू टू इम्बिबिशन से सीड कोट बिकम मोर परमिएबल टू ऑक्सीजन जो है वो एक्सचेंज हो जाती है ईजीली. आफ्टर इम्बिबिशन के बाद जो सीड है वो अपने वॉल्यूम में इंक्रीज होना इंक्रीज हो जाता है सीड, उसका साइज इंक्रीज हो जाता है, देयर बाय एक्सर्टिंग प्रेशर ऑन द सीड कोट लीडिंग टू रप्चर ऑफ द सीड कोट। समटाइम्स जब ज़्यादा प्रेशर सीड कोट पे पड़ता है तो सीड कोट विल बिकम रप्चर। नेक्स्ट इज रेस्पिरेशन। आफ्टर इनिशिएशन ऑफ जर्मिनेशन प्रोसेस, enormous energy is required for various biochemical changes which are met through rapid increase in respiration rate. Sucrose is probably the respiratory substrate at this stage which is provided by the endosperm. जर्मिनेशन प्रोसेस के बाद, स्टार्ट होने के बाद सीड को बहुत और केमिकल चेंजेस करने के लिए बहुत ज़्यादा एनर्जी की रिक्वायरमेंट होती है। तो ये एनर्जी की रिक्वायरमेंट कौन पूरी करती है? रेस्पिरेशन करती है। सुक्रोज इज प्रोबेबली द रेस्पिरेटरी सबस्ट्रेट है इस स्टेज पे जो एंडोस्पर्म प्रोवाइड करता है। इन ऑयल सीड्स एंड पल्सेस द लिपिड्स एंड प्रोटीन्स रिस्पेक्टिवली आर कन्वर्टेड इनटू सुक्रोज बाय सुटेबल बायोकेमिकल रिएक्शंस। थर्ड इज मोबिलाइजेशन ऑफ रिजर्व मटीरियल्स। As germination progress, there is mobilization of reserve material to provide. First is building blocks for the development of embryo. Second is energy for the biosynthetic processes. Third is nucleic acid for protein synthesis and embryonic development. Next are the biochemical changes. We have first is nucleic acid. हम फिजियोलॉजिकल चेंजेस स्टडी कर चुके हैं. नेक्स्ट इज बायोकेमिकल चेंजेस. इन मोनोकॉट्स ड्यूरिंग इम्बिबिशन देयर इज अ रैपिड डिक्रीज ऑफ डीएनए एंड आरएनए कंटेंट इन द एंडोस्पर्म with increase in the embryonic axis. Higher concentration of RNA precedes cell division. Due to more cell division, DNA content is increased. Next is carbohydrates. Insoluble carbohydrates like starch are the important reserve food for cereals in endosperm. During germination, starch is hydrolyzed first into maltose in the presence of alpha amylase and beta amylase, and then maltose is converted into glucose by maltase. The glucose is further converted into soluble sucrose and transported to growing embryonic axis. During germination, the embryonic axis secretes gibberellic acid into the allelonic layer and causes synthesis of alpha amylase. Third is lipid. Many plants like castor bean, peanuts store large amount of lipids or fats as reserve food in their seed. During germination, these fats are hydrolyzed into fatty acids. Fatty acids can further converted into acetyl by the process of beta oxidation, and this further converted into glycosylate cycle transported to the growing embryonic axis. Okay, next is protein. Some plants absorb protein as a reserve food in their seeds. Proteins are hydrolyzed into amino acids by peptidase enzyme. The amino acids may further provide energy by oxidation after deamination or may be utilized in the synthesis of new proteins also. And the last one is inorganic nutrients. A number of inorganic nutrients such as phosphate, calcium, magnesium, potassium are also stored in seed in the form of phytin. These stored nutrients are liberated during germination due to activity of various phosphates including phytase. So this is all about seed and their biological, biochemical and physiological